We're talking about this new study that came out, started to really hammer down into how our brains assign meaning to words. Okay, it's in the language processing part of our brain, and they had some really cool findings.
SciAnts. We get experimental science. We're curious, non-judgmental.
Let's talk about the brain. There's a really cool study that came out investigating the actual neural anatomy, how humans place meaning to words. So normally, a sound enters your ear. The auditory part of your brain can process the sound, but it can't assign meaning to the sound. It can just register and process the sound. Now, how your brain does this is a little bit of a mystery. There's been a lot of work into it, but we don't really have fine scale resolution. And I wanted to just explain to you kind of historically what has been done, why humans are the worst to work with, and then I'm going to tell you really quickly about the study because it's really cool. So now, Belant and I historically have worked with non-human models. Flies for our master's in graduate school for the most part. Stem cells like in petri dishes, sometimes mice, ants, as you know. We don't mess with humans. Occasionally I'll use human cells like in a petri dish. That's as close to human as I want to get. Because you're limited in what you can do. We don't want to hurt humans, even human test subjects. So it becomes very difficult to get detailed information, especially about how their brain is working. Listen, you can donate your body to science once you've passed, and that's great. If that's the right decision for you, you do it. It will help science. But you don't have living cells typically at that point. Or if you do, they're not intact the way we would need them to be intact. So if you want to understand how how your neurons respond to something, you're kind of stuck. You have limited techniques. Let me show you. I actually don't know what species this is. This is just a 2-photon microscope. That little bright spot that you're seeing in the center there, that's a neuron. And we can watch it in real time have an action potential. It means it's firing. It's sending its little electrical signal, chatting to neurons around it. Just watch this. Look at that. The pulsing. This is neurons talking to each other in real time. That's amazing, right? Can't do that in a human. There's a bunch of different ways you can do this with mice models and fruit flies. A lot of them involve genetic engineering. So we'll actually get individual neurons to fluoresce different colors when they're active. There's a lot of amazing things you can do. This is why mouse models, fly models are amazing. We don't have this resolution in humans. I'm sorry. Humans are boring to study for this reason. You can't get this for the most part. You can't get it. This is as good as we get when we're studying the brain for non-invasive stuff.
Andrea wants to know how that works.
How this
coloring or the flashing.
So I don't know exactly they don't give me a lot of details on this particular description. What it could be is probably a dye that's calcium sensitive, or so when there's electrical signaling, there's chemical and kind of like electrical gradient changes happening in the neuron that's pulsing, and so you can add in dyes, or you can genetically modify it so that you have a little light sensor, green or whatever, some kind of light sensor that responds to the chemical changes when the neuron is firing. Calcium sensors are one of the big ones, but there's a bunch of different ways you can do it. But you can track the electrical signaling, but it's extremely invasive. Like I said, a lot of times we have to gene edit things. So guys, so what we're studying, we're talking about this new study that came out that had, with some really beautiful resolution, started to really hammer down into how our brains assign meaning to words. Okay, it's in the language processing part of our brain, and they had some really cool findings. But I wanted to talk about why what they did was so extraordinary and why it hasn't been done to date, even though a lot of people have been studying it. And really, it's because we have a lot of amazing technology. Okay, I don't want to put down modern medicine. All right, it's great. But we don't have the finest map of the human brain, at least not while, the individual whose brain it is still using it, if you know what I mean. One of the best ways, we can monitor like brain waves generally with like EEGs, like lots of electrodes. The standard, I would say to date, is an fMRI. And there's this cute Alan Alda video that talks about fMRIs. And so, first of all, we do talk about fMRIs. I think it would be great for us to all like revisit what they are. But also, the past studies that were looking at the same thing relied on fMRIs, functional MRIs. So let's chat about that. And then I'll tell you about what the study did and why it's different than the past studies that were relying on fMRIs. So, fMRIs are cool, don't get me wrong. They're not giving us the resolution that we saw with the mouse or fly models where we can look at a single neuron, right? We have to watch blood flow. In an fMRI, it's like, okay, blood is moving to this region of the brain. And don't get me wrong, it can get pretty precise, okay? But there's still, dozens, hundreds, or even thousands of neurons in that cluster. You don't have the resolution that we get from animal models where you can watch what's happening to one single neuron and how it's talking to another single neuron that you're interested in. So this new study did something a little different. They partnered with a group that was going to be doing neurosurgery on a number of patients. They needed surgery because they had uncontrolled epilepsy. All of the medications weren't working. So this was all adults. None of the medications were working, so they needed to have a localized neurosurgery. And in order to do this, so epilepsy is like when you have unplanned brain activity, it can cause a range of issues, sort of from like small muscle spasms all the way to you can like the kind of classic lose consciousness body convulsions. It's from a sort of unwanted and uncontrolled neural activity, and so you can usually pinpoint the troublesome region. And so that's what they were going to do with these patients. So they were already going to have neurosurgery. As part of the neurosurgery, they have to have these little probes inserted into their brain to identify the very specific cluster of neurons that is the source of that individual's epilepsy. You see where I'm going with this? Huh? They already have probes in their brain. And conveniently, they were in the part of the brain that these researchers were interested in, the language processing part of the brain. Now, probes, you can start seeing how just one or two neurons are firing. Probe is you're actually measuring the electrical activity. And so this is extremely invasive. We do this for mice, less so for flies, but we do this a lot. Scientists do this a lot, but not for humans. It's incredibly invasive. You would never do this just for scientific study. But it's part of a medical treatment. They were able to piggyback on a medical treatment. So these patients already were going to have this probe in this part of their brain. They had already elected for this surgery, and then they were approached, hey, while you have this probe in your brain, would you be willing to listen to a couple hundred sentences and we can see what parts of your brain light up? Or like what clusters of neurons react. And so that's what they did. And they got like almost a dozen, I think, patients to do this. And they found something really cool. There'll be a handful of neurons, like, 3 or 6 neurons that respond to a specific word. But it's not just like you have a neuron that remembers the word munchkin and a neuron that remembers the word socks, the cat, right? That's not how your brain works, as it turns out. You have a couple of cells that will respond to a certain word, and they'll overlap. So one cell might respond to a couple of different words, but it responds to words that are similar. And I don't mean sound similar. It means similar in concept. So mouse and rat, very similar in a lot of ways. It's going to activate overlapping neurons. So it's not going to be the exact same pattern because our brain is able to tell the difference. But so it seems that meaning, our brains are actually organized to recognize things kind of based on meaning. Similar, it's programmed in our brain. Similar concepts are programmed similarly in our brain. It uses overlapping networks, kind of, which is really cool. And the other thing that's fun is these words were given in context. So they could be talking about somebody's son, like the family member. That lights up one region of the brain. But if you were talking about the star of our solar system, the sun, even though they sound the same way, and the auditory processing part of our brain would probably recognize those as the same word, the language processing part of our brain is like, no, these are completely different concepts. No overlapping neurons light up. It's stored in completely different parts of the language processing center. And, okay, the other thing that was really cool, they fine-tuned this enough that you could just, in theory, you could just look at the neural activity and get a pretty good sense of what the person just heard. It was almost like they joked in an interview that it was like approaching reading somebody's brain, but you're just like not very good at it. So they could get to the resolution of animal, action, place, animal, for instance, if that was the sort of structure of the sentence. They could tell that that person had heard a word that referred to an animal like mouse or cat, and that it was running, it was doing some kind of action, etc. And they could tell the order in which that individual heard the words. They couldn't necessarily say that person heard rat instead of mouse, but the brain clusters meanings in these general areas, so they were able to figure out what the different areas kind of do. And anyway, so the fact that when you hear a word, you do not have one neuron that is remembering, that is assigned specifically to that one word, as some of us joke. That is not what happens. But similar meaning words are stored in overlapping neurons, which is cool, and in the same brain region. And I thought that was amazing. Anyway, they'd kind of seen this before with fMRIs, but not at all of this resolution. They had electrical activity, they had neural activity from individual neurons, and it was beautiful. Okay, I was very excited about it, but I'm a neuro nerd, so. Saphira's mom, it would be interesting to see with people that have sensory processing issues or someone that doesn't have hearing. So what's interesting is that this is past the auditory processing part, it's the language processing. So I think if you could get people to read the word, or it's about like how meaning is stored. So in theory, I mean, it would be interesting, but I imagine it would be the same, even for people who couldn't hear, because they have other ways of, you know, engaging with language. And so the processing part of their brain would probably be the same, which I think is neat.
Do you know how deep was this? Are we very surface for the probe?
Surface-ish. Yeah, I mean, so in figure 1, the first figure 1A, they show you the placement of the probes.
There we go.
Yeah, so they do like neural pixels also, which don't matter, but what we're interested in is the red dots.
Okay.
So the red dots are the placement. So they're rather superficial. I mean, we're not talking about like center of the brain, but this is the language processing center, and deeper than little electrodes on the surface of your cranium.
Yeah, oh no, for sure. I mean, because it's touching the brain.
Yeah, no, it's actually inserted touching the brain, and it's able to track individual cell signals. So,
oh, this is cool.
Yeah, so this is neural activity, like the activity you're getting from an individual neuron in response to different words. So you can see if it's really small, it means there's no activity. If there's that big jump, that means the neuron is responding. So for instance, what we're looking at here, the didn't really have a reaction, but child had a big reaction.
This is the same neuron?
Yeah, well, they show you 3 different neurons.
But like in the little the upper graph
readout, yes. They show you the activity of 3 different neurons that were related, and you can see that they react differently. Mm-hmm. And that's what gives the specificity.
Yeah, so there's like a peak here on this one that's not there.
Yes.
And then maybe here is a little peak, but it's nothing like this peak.
Right, and so it's the overlap of a couple of cells giving the brain information that give it specificity, but the same cells are going to react to things that have a similar meaning, which is the thing that I think is bonkers.
Yeah.
Anyway.
That's really cool.
I thought it was neat. I was thinking about sending this paper, and I'm like, no, I'm going to get too in the weeds. I knew I was going to show you guys this. If I had this paper pulled up, and it really gets difficult to interpret. Like, listen, I love neurobiology. I spent a lot of my life doing the neurobiology thing, and it gets in the weeds. It can be hard to read these things, and I was like, I don't want to do it. But Belinda asked, so
It was all my fault, Chad.
I had to.
It's cool that it's semantic as opposed to phonetic because it implies there's some interesting top-down effect on the network level of individual neurons, so that neurons are in conceptual associations rather than purely sensational perceptual association.
Yeah, and also part of it is our brains react really differently to words in context. So words that sound identical. I gave the example of sun earlier. Am I talking about the family member or the star?
And those are two different brain regions.
Completely different brain regions, even though they sound the same, as long as context is given. So these words were delivered in a sentence, and so there was context, and sun and sun processed by different neurons. So it's the meaning behind it. And that was the question, is how does your brain store the meaning of words? Because the auditory system's been worked out. It's a little more clear cut now, but the auditory system. This is the cool stuff. How is detailed information encoded in your brain?